Hi, I'm Anisha. I'm a postdoc at the University of Washington, and today I'm going to talk about web-based neuroimaging tools for reproducible and collaborative research. So I think we've talked about many elements of reproducibility today, the first being study design. So I think we all know that we need to have a targeted, hypothesis-driven study that's well-designed, well-powered, that we're careful with data collection, that we plan our MRI acquisition parameters such that we get good quality data. Chris spoke about automation, how we need to standardize our computing environments. We need to decide on robust parameters for algorithms, and we need to define workflows. But I think the most difficult part about reproducibility is the human factors aspect. So we're all visualizing data, we're all exploring our data, and we're all making decisions based off of visual inspection. And this is particularly difficult to reproduce. So when I think about the human factors of reproducibility, I think of the algorithms that we're developing, and most of the focus has been on algorithms when we think about our science. But we also have to think about the interface with which we interact with our algorithms, and then we also have to think about the cultural aspects of reproducibility. So interfaces, they enable us to make decisions. So on the left, you can see a scientific data visualization of an MRI scan. I'm not sure if you can see, but if you look closely, it's very blurry, it's bad quality, and so the decision you would make is to remove this from your analysis. We also have information visualization, where you can see a scatter plot in the middle, and you can see very obviously there are outlier data points, and you would think, something's wrong with this data, I should look into it. We also use interfaces to read our papers. We read PDFs, and we make decisions on whether or not we want to include a paper in a meta-analysis, for example. So maybe you all are thinking, why is she talking about this? Ideally, algorithms would solve all these things. But as many of you know, algorithms can make mistakes, and we can't depend on algorithms for everything. Often these algorithms are trained by people, and so we need to find ways to make reproducible decisions. And so I believe the best way to do so is to design effective interfaces to our technology, and also think about cultural changes. Now that we have new technology, our culture needs to change around it. So my hypothesis is that web technology can help us perform more reproducible scientific research. And why am I talking about the web? Is it because I'm a millennial obsessed with my phone? No. The web is everywhere. Science is at our fingertips. You can access PubMed on your desktop, you can access Nitric on your iPad, and you can read a paper on your cell phone while waiting for the bus. So I'm proposing that we move from the wet lab, where we have a small group of scientists working on a small project, and we move to the web lab. And in the web lab, we can replicate and scale scientific decisions. We should use the web to communicate and collaborate more. We should use it to explore and share our data, and all of this will enable us to perform more reproducible science. So in this example, I'm going to talk about a project on reproducing expert decisions at scale using the web. So the objective here was quality control. The Healthy Brain Network is releasing up to 10,000 MRI scans in order to study pediatric mental health. And this is really great, but the problem is that kids move a lot in the scanner, as I'm sure many of you know, and these bad quality scans can affect our downstream processing and our statistical inferences, so we want to get rid of them. The problem is that automated QC is not quite perfect. And maybe some of you are thinking, well, deep learning, it's the new rage now. Sounds like maybe we should try it, but the problem is that you need labels, and you need a large set of labels. So we come back to the original problem. It's too many scans for one person to QC alone. So the solution, I thought, was to collaborate on the web. So I created this app called Mind Control, which stands for Brain Quality Control. And for those of you on your laptops, you can try the website right now at mindcontrol-hbn.herokuapp.com. As you can see in this video, you can open a nifty image. You can scroll through all the slices, you can change the brightness and contrast, you can pass or fail the image and give us a score of your confidence, and all of this is saved on a global database. There's even a leaderboard for you to compete against your friends. And so I showed this to a few neuroimaging experts, and they said, this is a really great idea, but Anisha, it's way too boring. I still don't want to do this. So this was a little frustrating, and I thought, okay, how can we present this data better such that I can get input from more researchers? So I redesigned the interface, and it's called Brainder. So for those of you who are bored right now, you can go on your phone, and you can go to brainder.us. So I made an application that instead of looking through every single slice of an MRI scan, you look at one single axial slice, and you swipe right if you think it's a good quality image, and you swipe left if it's a bad quality image. So I tweeted it, as you do, and I said, are you at work but feel like playing Tinder? Why not play Brander instead and help neuroscientists rate the quality of brain images? Swipe left to fail bad quality images. And I underestimated the power of social media, and this tweet became viral. I think I got 400 retweets. This ended up being nearly 95,000 ratings, so 95,000 swipes on only 3,600 images. So it was overkill. If you want to look at the preprint for the results, you can go to results.brander.us, but I'll summarize it here. The goal was to train a computer to emulate expert raters for the remaining HBN data releases. So Brander right now has data from the first 2 releases. And the method we used was to build this web interface to crowdsource this QC decision, and then we weighted the crowd by the subjective me, expert. And so this turned out to be very reproducible because the crowd labels were able to train a deep learning algorithm, and this matched the expert labels. So this was very promising. So now we have an algorithm that matches what my opinion would be. We could use it on the remaining HBN data sets. So then I was thinking, how would I replicate a study if I don't know how someone QC'd their data? I mean, this is really difficult. So in my preprint, you can see this interactive figure where I'm thresholding the data points of the first 2 releases, and I'm looking at gray matter volume during development. And I want you to notice at the top right the effect size, how it's changing, and at the bottom, the number of comparisons I'm making. So as useful as this web application is to interact and explore with my data, I am essentially p-hacking. And so this brings me to a data analysis culture change. You shouldn't play with your data, but you should explore it. And in order to explore it, you need to collect enough data to explore. So this means explore your data on the first 2 releases of HBN, but then stop. Make your parameter decisions there and don't play anymore. But also, I think in general, we need to be more transparent and share as much data as possible. I know some of you are working with PHI data, but it's really important to be as transparent as you can, especially when it comes to these sorts of decisions. So next I want to talk about a reproducible fMRI meta-analysis on the web. So as you know, we're starting to publish a lot. If you go on PubMed and you can look at the chart of fMRI publications, it's increasing exponentially. And so ideally, we need to run meta-analyses to understand our results and make sense of the thousands of publications that are happening. So we built this app called MetaCurious, and if you're on your computer, you can visit it now. And the goal is to keep track of articles and metadata in an fMRI meta-analysis. So as a background, NeuroSynth is an application that extracts activation coordinate tables, but not effect size. And for some meta-analysis statistical methods, you want the effect size. But the problem is that different papers report effect size in different ways. So ideally, we need manual annotation. So what we thought was, let's build an efficient web interface to annotate the effect size table. And this would be reproducible because all annotations would be version controlled through GitHub. And this is important because for those of you who've done a meta-analysis, you know that you always make a choice on which paper you want to include in your analysis or not. And when you decide not to include a paper in a meta-analysis, your reviewer might ask you, well, why did you drop that paper? And so version control of a meta-analysis is really important because then you can go back and say, oh, this is why. So in this video here, I'm searching for a topic. I typed in memory. I'm opening the first link. We open to a split screen view with the PDF where you can scroll and zoom into the effect size table that you want to annotate. Okay, so that part looks good. Okay, we can see that these are MNI coordinates and they're reporting the Z value, so I'm annotating all of that. I scroll down to the effect size table and I can start typing in the Z-scores. And so this is a collaborative project with Katie Bottenhorn, Elizabeth Dupree, JB, Neil Sharvesh, and Greg. But I kind of lied to you just now. It's actually broken, and this is because of paywalls. So I thought that the best interface to effectively annotate something from a paper was a side-by-side view. But it turns out that publishers don't like it if you look at a paper not on their website. So here I am pleading to you to please publish openly. At least put your paper on bioRxiv so that your data and your science can be part of this bigger picture. If I can't access your paper, that means I can't annotate it, and that means it's not part of a meta-analysis. I also think we need to publish less, slow down a little bit, collect larger data sets, do more collaborations. Oh, Okay. And I also think, after you've slowed down, you should still publish your data and your results regardless of negative results. Because if we only publish things that are giving us results, we're going to have a biased meta-analysis. So to conclude, the interface between scientists and algorithms is an important factor for reproducibility. I think the web is an untapped resource for science, and that it could really help us perform more reproducible research. But I also think that technology is not the full solution to the reproducibility crisis, and that it requires a cultural change. So with that, I'd like to thank all of my collaborators, and especially everyone who's rated images on Braindr, and my funders, the UWIN and the UW eScience Institute. And feel free to tweet at me on Twitter, or email me if you have any questions.